Good morning. Today we have Dr. Prakash. Presently, he is working as a principal scientist, Analytical R&D Center, the Himalaya Drug Company, Bangalore. He has done his SGM College of Pharmacy, Chitradurga, MPharm from Pharmacy, and PhD from Rajiv Gandhi University of Health Science, Bangalore. He has vast experience of R&D. He has 20 years experience in analytical R&D in nutraceutical, cosmeceutical, and pharmaceutical. Presently, he is working as a principal scientist and overall in charge of all activities in ADL at Himalaya Drug Company. He is 16 in analytical R&D. Hello. Presently looking after analytical R&D 6 teams like a phytopharmaceutical team, pharmaceutical analytical team, functional food analytical team, cosmeceutical active analytical team. So he is the overall in charge of analytical R&D. He has developed team for literature survey, identification, selection, method development, review, and finalization of phytomarkers in pharma, functional food, and AHP products using instruments like HPLC, GC, MAS, etc. His major achievements are like establishment of complete toxin portfolio like mycotoxins, heavy metals, pesticide residue, etc. are becoming mandatory for all the products. In addition, AHP products need special requirements in many countries, and he has established all the analytical platform required to meet this global requirement. He has developed seasonal and geographical variation, a systematic science-based approach has been initiated to understand challenges, evaluation and study the impact of seasonal and geographical variation on the quality of medicine. Please mute yourself. Please mute. Develop analytical method. He has done the CTD dossier, preparation of CTD dossier for various pharmaceutical formulations to sustain the registration in various countries. He has done for that 12 products. He is instrument for in-house training as well as for external delegates. He has collaborative research work, has guided 10 project trainees from PES College of Pharmacy, Swami Vivekanand College of Pharmacy, and some other colleges, and he has guided successfully research scholar. He has worked on herbal formulation stability studies as well as degradation of antioxidants in cosmetic preparation. He has innovated development of herbal eye drop Opthacare, analytical method development of Opthacare, and its stability studies by gas chromatography. He has participated as a resource person at various universities and colleges like PES College of Pharmacy, Bangalore, KT Patil College of Pharmacy, Maharashtra, KLS Ayurveda College and Hospital, Belgaum. Then he has also delivered lecture at Vinayaka Mission College of Pharmacy, Tamil Nadu. He has attended conferences and symposium. He has published papers in national and international journal. He has received Best Paper Award, and he has developed 15 monograph for United States Pharmacopoeia. Now I request Dr. Prakash to deliver his lecture.
Hello?
It is audible, sir.
Hello?
Sir, it is audible.
Yeah,
Sir,
okay.
screen is also there, means your PPT is also there. Okay, sir.
Okay, thank you. Good morning, everybody. Can you hear me?
Yes, sir. Good morning.
Okay.
Yes, sir. Very good morning.
Good morning. Good morning. Respected Principal Dr. Jalal Prasad, Vice Principal Dr. MB Patil, sir, and HOD and Professor Dr. Kalpana Madam, Dr. Pramod, sir, and all faculty members and all participants. Myself, Dr. NS Prakash, working as a principal scientist in R&D Himalaya Drug Company.
This conference will now be recorded.
I'm having almost 20 years. I've been working in the Himalaya Drug Company in various segments. Now, it is very proud for me to participate as a resource person in the quality improvement program. Even in the year of 2001, 2002, when I was as a student, even I participated as a student. That was the first quality improvement program was conducted in the KLD College of Pharmacy. Now I am very happy. After 20 years, I am participating as a resource person. My topic is Phytopharmaceuticals, Challenges and Opportunities. These phytopharmaceuticals we have been telling from the so many years, but now it is different from earlier. If you go to next slide. This now India's strength in herbal technology. Herbal, because India is from the last so many years, from the 500 years, we are leaders in the herbal technology, because in the ecology of India, more than 50,000 plant species are documented. In the Indian flora itself is more than 18,000 plants are documented. The Indian flora is very rich because it is about around 6 to 7% of the total flora of the world is coming from the Indian flora. If you talk Indian traditional medicines, there is Ayurveda, Siddha, and Unani, and modern medicines. Though there are more than 18,000 plants, these species are documented in the Indian flora, and only 8,000 of medicinal plants are used by the tribal peoples. Though the 8,000 species are used in the tribal people, only 900 species are documented in Ayurveda, and 700 species are documented in the Unani, and 600 species are documented in the Siddha. Even modern medicines also, whatever the molecules available in the plant, they are isolated and used in the pure form in the modern medicines. Now, why that herbal technology or herbal industry is very, very important in the latest days, because these herbal drugs is not only used in the pharma, it is used in the natural foods or nutraceuticals, and even it is used in the cosmetic industries. If you see the projections of herbal drugs growth, it was 2009, it was only $62 billion. Now in 2020, it is a 5 trillion. It is almost 80-fold increase in the growth market in the herbal industries. That's why there is so much of importance in the herbal technology or herbal products. If you see that, what is the next alternative medicine for the modern medicine is herbal medicines. In the herbal medicines, we are finding one common terminology is a standardization, and other one is a medicinal substances, medicinal preparation, and medicinal product or herbal drug. If you see that herbal substances, it is a mainly whole or fragmented or any part of the plant or algae, fungi as unprocessed in a dried or fresh form. If you take herbal preparations, any preparations or any extractions which is prepared from the herbal substances is called herbal preparations, either by the doing extractions or distillation or expressions, fractionations and purifications. Herbal medicines is herbal drug is any, either we can use as a herbal substance powder or we can use as a herbal preparation. From these two actives we can make herbal product by using these two, either herbal powder or herbal preparations for the diagnosis, cure, mitigation, treatment, prevention of diseases in man and other animals. Now, why the phytopharmaceuticals is very important? We have been phytomolecules, we have been doing research from the so many years, but now in the current days, phytopharmaceuticals is having very importance. First of all, we should know what is phytopharmaceuticals drug. Phytopharmaceutical drug, in earlier days, we academicians and research organizations or research institutes, they were doing that research on by using the botanical drug. They are doing that extractions, fractionations, and even they were doing that bio-guided fractionations, lead identification of the lead molecules and their purifications. They were identifying the molecule responsible for the activity, but even then the question always arises, what next? Because they cannot go to that commercial level, because they were not having the platform to explore their research. Now, in the government of India, they passed the Gazette notification in 2015 for the formulation of phytopharmaceuticals. They were so far in the 2008 they made the committee for the what are the problems facing in the if you introduce phytopharmaceuticals. For that they have made a committee. The committee, they have done the review all the things and they recommended it is not having that overlap with the overlap with the Ayurveda and Siddha and Unani. Now they have made the central government to understand. Now after that it is passed in the Gazette. Now what is the phytopharmaceuticals? Phytopharmaceuticals is a drug include purified standard fractions with a defined minimum 4 bioactive or phytochemical compounds qualitatively or quantitatively assessed for the extract of medicinal plants and its parts for external use or internal use by human beings or animals for diagnosis, treatment, prevention of disease or disorders, but does not include administration parenteral route as specified in the Rules 2EB of Drugs and Cosmetics, 1995. Here, any phytopharmaceuticals we can develop, but make sure that either 4 bioactive constituents or phytoconstituents you have to identify qualitatively or quantitatively. That is the minimum criteria in the phytopharmaceuticals. Why phytopharmaceuticals is required? Here, first one is plants-based research fraction to be used as a drug. Suppose if you see that at present in Ayush, Ayurveda, Siddha and Unani medicines, there is no scope for the fractionation on purification process. It is not allowed. Because of this, now even in phytopharmaceuticals, we can use by using various solvents, we can make it to extract it and further we can fractionate it and we can purify it to single molecule. And those molecules we can evaluate either as a biomarkers or phytomarkers. Same thing we can identify in the finished product, we can quantify. This is possible at present in the phytopharmaceuticals. Next one is science-based drug for botanicals of traditional medicines. Whatever the traditional medicines we have discussed, either Ayurveda, Unani and Siddha, same ingredients we can take it, same formulation we can take it, and we can do further research and we can give the science touch for those formulations, those ingredients. But so far in Ayurveda and Siddha and Unani there is no scope. But in phytopharmaceuticals, science-based research is possible. Herbal drugs are poorly regulated and controlled because in Ayurveda, Siddha and Unani, it is trust everything, because any formulations, if you prepared and processed as per the textbooks, as per the procedure given in the textbook, the same formulation you bring as a product, but there is safety regulation is not required, clinical study is not required, directly you can use as a product. That's why these herbal drugs so far it is not regulated properly. Now in the phytopharmaceuticals there is a very good scope for the regulations and very good scope for to bring that more and more guidelines. Now encourage the interest and demand of the plant-based therapeutics out of Ayush principles. Because of this reason, even academic institutions and research organizations and research institutes, there is a scope for to encourage the interest and demand for the innovations. Unlike conventional medicines, suspect everything, and Ayurvedic medicines trust everything. Because this phytopharmaceutical acts as a bridge between the conventional medicines and the Ayurvedic medicines. History of phytopharmaceuticals. Some of the synthetic molecules as they emerged out of their quantitative structure activity relationship. Because from the long time we have been working in the herbal drug, some of the research institutes, some of the drugs which is explored as a single molecules. If you see that quinine, physostigmine, codeine, salicylic acid, these compounds are evolved in the research of herbal drugs, and they are still using in the modern medicines. Because of this reason, there are in various countries still they are working on extractions, fractionations, and further fractionations and purified fractions. The same kind of activity we can do in India also. Phytopharmaceuticals versus modern medicines. The conventional medicines suspect everything and Ayurvedic medicines trust everything. I already told conventional medicines, because of having the high toxicity, always each and every molecules, though we are saying is safe, always we are suspecting the drug for the long treatment. In Ayush medicine also, since these are all from the long time using, and more millions of people they use Ayurvedic medicines, they are trusting it is always safe drug, but it is not true. Next one is toxic effect of the various synthetic drugs has forced research academicians and doctors to consider some steps for preventing the misuse of such drugs. Because of modern medicines are very toxic in nature, this leads to the explore the phytopharmaceutical drug, which is, since we are always telling that herbal medicines or herbal botanicals are not toxic, now we can explore the phytopharmaceuticals and their possibility, toxicity and pharmacological activity. Next one is occurrence of the side effects after long-term usage of synthetic drug is always feared during the treatment of chronic disease. This I already told. Because of this modern medicines having the toxicity, this lead to the evolution of the phytopharmaceuticals. Next, phytopharmaceutical versus traditional medicines. How does phytopharmaceutical drugs differ from the Ayurveda, Siddha and Unani under section 3A and H of Drugs and Cosmetic Act 1940? Because these traditional medicines, Ayurveda, Siddha and Unani, are manufactured exclusively accordance with the formula described in the authoritative books of Ayurveda, Siddha and Unani system of medicines specified in the first schedule. I already told, because since if you are preparing any preparation of any product according to the textbooks, using the ingredients used in the text and amount of ingredient, what quantity of ingredient present in the formula, if you prepared as per the Ayurvedic test, then it is considered as we can register as a product in the Ayurvedic principles. But that is in phytopharmaceuticals, we can do the extraction, we can do the fractionation, and we can do the further purifications, and we can identify the leaves. That is possible in the phytopharmaceuticals. Patents and preparatory medicines makes use of ingredients referred in the formulation of authoritative text. Since as per the Ayurveda, we have to prepare as per the formula given in the text, then patents and proprietary medicines make use of ingredients in the formulation of authoritative text books. However, the phytopharmaceuticals, drugs and fractions of crude drugs are distinctly differentiated by using purified standardized with the minimum 4 bioactive or phyto compounds present in the either qualitatively or quantitatively. That's why phytopharmaceuticals now it is going to be new modern drug in the herbal industry. Requirements for the submission of the application. Suppose if you wanted to submit the application for the either to manufacture the phytopharmaceuticals, or you wanted to import the phytopharmaceuticals, or you wanted to do the clinical study of the phytopharmaceuticals, well, you have to apply the application to DCGI. While applying, what are the documents you have to give along with the applications? There are two categories of documents. One category is whatever available in the existing literature. Whatever the available, no need to develop once again. Whatever is available in the existing literature, those documents, part one. Other part two is if the existing as per the literature, documents are not available, you have to create the document in part two. First one is whatever the whatever available in the literature, we can submit directly. That is description, summary of the phytopharmaceuticals, drug or botanical name, formulation, route of administration, dosage, indications, and claim. Even whatever the available in the literature of any botanical drug, you will be submitting the document of what is the biological source, what is the family, and their macroscopic characters, microscopic characters, and authenticity of the botanical, from where you are cultivating, where you are collecting, whether you are following that good cultivation practices or good cultivation and collection practices, and what is the season you are collecting, and what is the geographical location you are cultivating. All the documents we have to submit along with the application. Next one is published literature and traditional medicines or ethnomedicines and reference book. Whatever, when you are selecting the phytopharmaceuticals, from where you are taken, whether you have taken from the traditional medicine books or traditional medicines, or if you have taken from ethnomedicine or any reference book, from where you have taken, and their composition, dose, method of usage, and active ingredients preparation, how we are preparing that, and what is the dose of phytopharmaceuticals. Complete information you have to submit. Next, information on any contraindication or side effects. Suppose if any contraindications or side effects are reported as per the literature, that also you have to disclose. And published scientific reports, safety and pharmacological studies. Even in some, in part 1, no need to create any document of safety and pharmacological activity. If any your drug is already reported, anybody has reported, and already the request safety studies report is already available, and pharmaceutical studies are already available, the same study we can submit along with the application. Next one is present usage of pharmaceuticals, drug, and history of usage and of the product manufactured, quantum of sold, extent of exposure on human population, and number of years. See this, if any manufacturer already he is having the product, and if we could able to prove minimum 4 bioactive molecules or 4 phytopharmaceutical molecules present in his existing formulation, he can submit directly without any data. But along with the application, he has to do that, because safety study may not be required, because based on the history of usage. Suppose if the product is already in the market for 10 years, then so many people are, how much of market, quantum of market you have to, and make them to understand, and what is the volume of the market, how many units he has sold, how many years he has sold, and how many population he might have taken. Based on that, all the document he has to submit. If they are able to, any manufacturer is able to satisfy the regulatory people without any soft safety and pharmacological data, he can do the registration. Other one is human and or clinical pharmacology information. If it is not available, then published this scientific reports of pharma studies, including human studies or clinical studies, you have to submit along with the application. And pharmacodynamic study information is available, that also you can submit. And any monograph, published monograph in the various pharmacopoeia is already available. Same thing you are going to consider as a phytopharmaceuticals, that also we can give as a document along with the application. Next is, if any data is not available in the literature, we can generate the data in the part 2. If you wanted to generate the data, same thing, whatever the data is submitted in the application in the part 1, the same thing you have to create and submit. That also possible. Identification, authentication, source of the plant, determination of endangered or threatened under CITES, any known, whether it is a botanical drug, is a genotypic, chemotypic, or ecotypic, what variant, and if you are growing that botanical drug, where you are growing, who is growing, what geographic location you are growing, what climatic condition you are growing, how is the cultivation, and their quality parameters, specifications of including all the parameters, and the washing, drying, and extraction process and fractionation process. The complete data we have to submit along with the application. Next, what are the extracts you are making? What are the fractions you are making? How we are characterizing how we are purifying it? How we are characterizing using modern hyphenated tools? And how the bioactive constituents or analytical markers you are identifying, that characterization you have to complete document you have to give, and inform if you are making that finished product, what excipients you are using, what your diluent you are using, that complete data we have to give along with the formulations, and even composition of the formulation with the defined markers you have to establish, and you have to give the document. And manufacturing process of the formulation, you have to give the complete MFR, means manufacturing record, you have to give it with what instruments are using, what are the instruments ID, how we are processing, how long mixing, drying, and complete formulations, and along with the stability data of actives, and also as a finished product as per the latest ICH guidelines. And even finished product also, details of the packaging of the finished product, and even along with the quality control identification and quantifications, qualitative specifications of actives and inactives, and since it is 4 bioactives and 4 phytomarkers are mandatory, you have to give that chromatographic fingerprint profile along with the phytochemical reference markers and its assay. And since safety data or pharmacological data, if it is not available in the existing literature, then we have to establish the safety data and pharmacological data as per the guidelines. And you have to carry out the human studies and clinical trials, trials as per the latest guidelines. What is the opportunities? How opportunities creating? Crude drugs are subjected to the suitable method of extraction, fractionation, and purification for the isolation of phytopharmaceuticals, which could be incorporated as an active ingredient in the modern system of medicines. Because I already told, in Ayurveda, Unani, and Siddha, there is no scope for the extraction and fractionation and purification. But in phytopharmaceuticals, there is a scope for you can use any solvent and use for the extraction, and we can go for the fractionation and purification, and we can go for the purified compound level. That same thing we can incorporate it into your product is equivalent to the modern medicine. That possibility in the phytopharmaceuticals. By this process, promote the innovation and development of the new drugs from the botanical in scientific way. Because any botanical drug, if you are taking, you can give the scientific touch in the identification and quantification of the biomarkers, so that we can prove that what is working, what is not working, and if it is working, means how it is working. That we can give in the phytopharmaceutical drugs. Next one is encourage to research in the drug development for innovators, academicians, and industry and national laboratories. So many, even you academicians, now we can take that any drug, any project is having the scope, because now if you take any botanicals, you can take one student and we can do that extraction and fractionations, and we can identify what fraction is active, which fraction is active by using that pharmacological study. With one student you can do it, and next student we can continue the same fraction for the identification and characterization. Now it is very good scope for the academic people which can work on phytopharmaceuticals. Earlier days it was not like that. If any academicians work, he is working only for the publication purpose, because he has to do the research and he has to publish it, and he has to take his degree. Only that much it was available. Now he can go further and he can do that commercialization of whatever his research. Now you are academicians, we can take the project and we can do further in different students, and we can reach that with all biomarkers level. This is very good opportunity for academic people. As a industry, always they will always run with the timeline. It is very difficult to do it. If any academicians, if you are doing it, and we can same product we can sell to the industries. And by this is also giving the opportunity to use the modern techniques, because in the Ayurveda it is not possible to use any solvent other than alcohol. Okay, but in the phytopharmaceuticals, whatever solvent you can use it, either chloroform, ether acetate, or acetone, or ethanol, or methanol. But you have to make sure that whatever the residues which are present in your active extract or fractions or finished product meets the residual level of solvents as per the ICH guidelines. That we have to make sure. But there is a scope for the use of various solvents and fractionation method and potentiating the fractions that is possible in the phytopharmaceuticals. Next is phytopharmaceuticals, not only from the Ayurveda, Siddha and Unani, but we can use any phytobotanical drug, not only from the Ayurveda, Unani and Siddha we can choose it, but other medicines, campo medicines, botanist medicines, and we can take any botanical drug from the ethnobotany, and any drug which is using the tribal practices and other sources we can take and we can do the phytopharmaceuticals. But earlier, if any drug, if you are taking from the out of Ayurveda text, it is not allowed to register in the Ayurvedic proprietary medicines. But in the phytopharmaceutical, that possibility we can explore. Then effective utilization of the modern technology for the characterization. Because here though, since the identification and characterization of minimum 4 biomarkers and phytomarkers is mandatory, then we can use the hyphenated technique. Like MAS, NMR, HRD, and whether these all hyphenated technique, we can use it for the identification and characterization. And this safety and efficacy data can be established, because since Ayurveda is trust everything, there if you are any preparation, if you are making as per the Ayurvedic principles, there is no need to establish the safety and efficacy data. But that's why here present in Ayurveda, we are not finding so much of data of safety and efficacy. But in the phytopharmaceuticals, we can explore the safety and efficacy of the each and botanicals and even molecules present in the botanical. That possibility it is allowed. And whatever the phytopharmaceuticals we are developing, it is approved by the DCGI, would have the same status of market as equivalent to the synthetic drug. Whatever the molecule or whatever the molecule you are developing phytopharmaceuticals, it will be equivalent to the status of modern medicines, since it is approved by the DCGI, Drug Control General of India, and allowed to prescribe by both MBBS and BAMS qualified physicians. In Ayurvedic medicines, already as per the latest verdict from the court, MBBS drugs, MBBS physicians should not prescribe Ayurvedic drugs, and Ayurvedic BAMS qualified person should not prescribe modern medicines. That differentiation is present in the existing. But in phytopharmaceuticals, it's an opportunity to have both. And one more is new entrants, innovators, or startups will get encouraged to invest in the phytopharmaceuticals. Challenges and the limitations. What is the challenges? Because since we have to create so much of data for the registration of phytopharmaceuticals, But moreover, one minimum 4 bioactives or minimum 4 phytoconstituents you have to identify and quantify. But because of this reason, small and medium enterprises do not find it is cost effective. To do all the things, it is so expensive. Because of this, small and medium enterprises, it is difficult. And clinical trials, international patents, and product registration for international market is a major cost component, because if you want to register any product in the international market, because you have to undergo the safety and clinical trials. Each clinical trials is so much of expensive. That's why registration of this product is expensive. Due to the lack of regulatory system, very little scientific work can lead to the development of rational evidence-based phytopharmaceuticals, because still it is very premature. There is a lack in the regulatory system. Is it going forward, we can understand and we can do more and more as per the regulatory requirement. And present unfriendly patent and related regulations in the systems. Since it is a very premature, we don't know that what is the patent, what is the regulations, and still we have to, going forward, we can understand more. That's why these phytopharmaceuticals, whatever the quality parameters, what is standardization parameters, we are following for the existing herbal products or herbal ingredients or herbal preparations. The same quality parameters we have to use for the phytopharmaceuticals also. Same color, odor, taste, texture already discussed is then microscopic characters, and qualitative, quantitative identifications of markers, and functionality related. If it is a product, moisture content, disintegration test, weight variation, friability, dissolutions, and then if it is a botanical ingredient, extractive value, ash value, and then chromatographic fingerprints. Is it a qualitative and quantitative? And heavy metals, pesticides, aflatoxins, and bacterial and fungal, microbial contamination, and toxicological data, pharmacological data, and clinical data. Whatever, these are all is required for the registration of the phytopharmaceuticals. These are the organoleptic, botanical, physical, and chemical and biologicals. Since it is a phytopharmaceuticals, we always, when we are doing the research, we have to do the, what is the end point. From our focus towards the research is, first we have to identify minimum 4 bioactive constituents and minimum 4 phytoconstituents. Based on keeping in those things in mind, we have to do that extraction, fractionation, and we have to do the pharmacological activity and toxicity, and we have to purify it and identify and quantify using the hyphenated techniques. This I told no identification. If the markers are already available in the literature, we can identify those markers as per the literature, and we can identify and quantify. If it is not available, we can develop your own, and by identifying the major markers as per the TLC or HPLC. And if it is not available in the literature, and you have to further fractionate it, isolate by using the column chromatography or preparative chromatography, and you isolate each and every molecules, and you have to characterize those molecules. These are the, you can see that how all 4 compounds you could able to identify in the extract level, that those compounds are purified in the purified form as a single entity. Those things we are just characterized. The same format concept you use for the each and every botanical. Now I'll show one example how we have to develop one botanical in line with the phytopharmaceuticals. One example I'll show. This is now example I have taken is a neem phytopharmaceuticals. You know that neem is very well known drug, is commonly available and is widely used in the Ayurvedic industry. And the same neem, if you do development in line with the phytopharmaceuticals, how? I already told everything regarding the availability of the source, cultivation, collection, macroscopic and microscopic characters, and geographical location where you are cultivating and what season you are collecting, all the details you have to submit. Along with that, macroscopic characters, you can see that macroscopic characters, and you can see that microscopic characters of neem leaf that already you are doing in your colleges. No need to explain, you are expert in those macroscopic and microscopic characters. We also, industries, we are doing that same, whatever you are doing in the laboratory, the same thing we are doing, and we are submitting for the regulatory requirement. This is the microscopic of epidermis and TS of the petiole of epidermis, and this is the seed of Azadirachta indica, macroscopic characters, and seed of microscopic characters of Azadirachta indica. Even powder analysis also we are doing in the industry. Thus, whatever you are doing in the laboratory, the same way we are doing in the industries, and we can identify the uniqueness of any botanical drug. And I already told, whenever you are developing any phytopharmaceuticals, you have to keep biomarkers or phytomarkers in the mind. And before you are doing that any research, first you have to do that complete literature search of these phytochemicals. This, if you take the examples of neem, it contains, there is very much, so much of work is done on the neem. There is so much of published literature available in different domain. There, if you see that phytopharmaceuticals, there are various group of phytopharmaceuticals is reported in neem. Mainly is limonites is the major compounds. Along with the limonites, it is flavonoids, catechins, triterpene, and gallic acids, and phenolic acids, and azadirachtins, and so many. Even if you see that nimbins or belongs to limonites, is the major components. Then whenever you wanted to do any phytopharmaceutical drug, we have to do complete literature search, and you have to understand that structure of the each molecules, each groups, and what is their content. If already reported, you have to make them to understand. And you have to understand what is the structure of the each compound, how many sugar moieties are attached, whether it is a free form or glycosidic form, and whether it is in flavonoid group, what are the different class of flavonoids which are presented, and their structures, and their mass, and their UV spectra, and you have to, in depth, you have to understand. And if at all any literature is available for any identification and quantification of the phytomolecules, then we can take those literature, and we can perform in-house, so that it will give that value. We can take that different parts, was a raw fruit or leaves. Now we have taken that leaves, bark, and fruit, seed. Then we have done that in-house. And even literature also, doing that, if you wanted to identify the biomarkers, we have to do the literature about where and all it is using, and what for it is using, and then you can, based on that, we can do that subjected to the extraction, fractionation, and bioguided fractionation subjected for the activity. This already told you have to identify the different structures of the different compounds, and you have to understand what is the side chain, how it is different side chains are attached to that main group of main basic structure, to understand during the identification and the quantification. And you should understand that what is the chemical name, what is the molecular formula, and what is the molecular weight, and so that further mass-based identification it will be easy. And you should understand for NMR, if you're submitting for the NMR, so that these are the literature data is required for the identification and characterization. Then during the development, if all the literature data and their structures and then mass value formula, everything is available, all the data is available, you can do some HPLC method and you can run the whatever your extract or fraction or product by using
This
HPLC.
conference will now be recorded.
Now we can establish the chromatogram so that we can identify, if you run any HPLC, we can find this kind of chromatogram. From this we can understand, based on the literature, if you are having the complete literature, we can understand that these are the bands which belongs to the phenolic acids, these are bands that belongs to the flavonoids, or it belongs to triterpenoids or limonides, so that we can understand based on their structures and their wavelength, lambda max, and so that we can tentatively we can identify what could be the different bands. Based on the literature, now you have different nanometer name, you can see that these are at 225 nanometer, you can see that phenolic acids. The same thing, if you go to the 200 nanometers, these are the compounds, could be the limonides. Okay, the same way we can do the literature and we can predict the compounds, what could be the compounds based on the available literature and their spectra and lambda max value. See that the same extract, if you do, if you identify, you could see at 254 nanometer, we can identify the flavonoids. And the same extract or fraction, whatever we are having, we can subject for the LCMS. And the LCMS, we can see that LCMS chromatogram, finding the various bands and at different nanometers, whatever at 225, 325 and 354. After LCMS, we can find out the different masks of different RT. Based on that, we can predict. You can tentatively we can predict at the LC level, HPLC level by using UV spectra. The same thing we can confirm by the mass. And further fractionation also we can do it in the LCMS, so that we can fragmented mass we can identify and we can characterize the what is the molecule you are looking. So after that we can match to compare with the literature mass, and we can identify what could be your peak of interest, and whether whatever predicted in the HPLC, whether it is the same in the mass or not. Then further, suppose if you wanted to do further characterization by using a NMR, you can compare with the data, all the data, whatever you established, you can compare with the literature. So that now, based on the UV spectra and mass, we can finally predict the earlier you have predicted only group of molecules. Now, based on the UV spectra and the mass spectra, mass value, we can, and fragmented mass, we can predict what are the different compounds. Now we can see that identification and quantification of flavonoid markers in the neem leaf. Then, if any mark after the identification and characterization, if the standards are available commercially, or you yourself are isolating and characterizing the markers, then we can run we can create as a reference standard, And along with the reference standard, you can quantify the how much it contains. Now we can see that routine along with the available standard routine and nimboloid we have isolated and characterized. Along with the nimboloid we have identified and quantified by using HPLC in different batches. And apart from that routine and nimboloid, we have quantified D-acetyl nimbin and D-acetyl selenin in the different extract and fractions. This is the nimboloid and the D-acetyl nimbin and D-acetyl selenin identified and quantified in the fraction. From this, we can quantify what is the content of nimboloid in the different samples and D-acetyl nimbin in different sample, D-acetyl selenin, and cumulative of all is we can call as a limonides. These are the along with the identification and quantification of the phytomolecules. We have to do other parameters also: description, LOD, total ash, acid insoluble ash, water extractive value, alcohol extractive value, and the HP. And along with the assay, we have to do the TLC also. Once again, that quantification of different parameters and assay and TLC. Even not only that botanical, whatever the intermediate also we can, whatever the intermediate are using extracts or fraction, we can identify and quantify. This is the name of extracts and fractions that we have done that HPLC and quantified in different batches, nimboloid and routine. See that what is the content of routine, even more than 1% routine present in that. Nimboloid is 0.55 in the extract level method, that is neem methanol extract of different batches. One is 0.55%, other one is 0.97%, other one is 0.42%. Apart from that quantification, as we told, we can do the identification also. Identification also we have to possible in the neem. If I am giving one trial, whenever you are getting any botanical or any extract, you have to run that chromatogram by using different solvents, UV 254, 366, and white light. This neem I have performed in one trial. We can see that other trial also. Based on the resolution, you can finalize the what method or what solvent you want to do it. Next one is identification. You can see that identification with respect to the marker. Nimboloid marker I used the different batches of neem leaf. Okay, you can identify that nimboloid marker in the as per the TLC. Is one more different batches we have performed. We can see that beauty of the TLC, how it is uniform fingerprint as unique uniform of different batches. Okay, these are the uniqueness in the herbal botanicals. And identification of the routine by TLC, we can see that you can see in UV also, and we can see in the fluorescent routine that we can find out in the different batches. Next one is identification of routine, same routine in different batches. Now identification of D-acetyl nimbin and D-acetyl selenin and nimboloid in different solvent system. You can see all the three markers you can identify. Now nimbin, nimboloid and azadirachtin identification by TLC. These are all different solvent. You can try different markers. You can try, and so that identification and quantification you can perform. Routine quercetin of different all flavonoids, 3 quercetin 3-O-glucoside and the quercetin 3-O-glucuronide. From this, we can conclude this phytopharmaceutical is a very good platform for to showcase whatever the research you wanted to do it. Whatever the research you wanted to do it is a very good platform you can do the research, especially in the academia who are very much interested to do the molecular level research. Now that we wanted to make it to applicable for the commercial level, we can take to the commercial level. It is a very good platform. And we can create the roadmap to explore the traditional medicine to scientific way. Because I already told, by AICTE it is very less scope but in phytopharmaceuticals we can establish the roadmap to explore the traditional medicines in a scientific way and explore the modern analytical tools or technology in the development of pharmaceuticals, because it is allowed various solvents and various processes. We can modify the process. We can do that CO2 extraction or fluid bed drying or various technology. We can use it to. This is very much possible in the phytopharmaceuticals. And even if you wanted to characterize the molecules by various tools, you can use hyphenated tools. You can use this for LC, NMR, LC, MS technology for the characterization of the phytomolecules. And possibility to answer the what is working, how it is working. Because in AICTE, it is difficult to tell what is working and how it is working. But in phytopharmaceuticals, there is a possibility you can tell what is working and how it is working. And markers-based biomarker identification and quantification of herbal substance preparation in the phytopharmaceuticals. There is a possibility to whatever the traditional medicines or ethno-medicines or tribals which are used, those botanical drugs we can make it to modern medicine. That with all this, I'm going to conclude the importance of phytopharmaceuticals and how we have to develop and how we can take the bench to bottle level. Thank Jalal Professor, M.B. Patel sir, and Kalpana madam, and Pramod sir, and all faculty members and management of KLE giving me an opportunity to explore or showcase our industrial research to all the faculty members. Thank you one and all. Hello?
Yes, sir.
Before we thank Sir, the session is open for any doubts and queries, clarifications. If any of the participants have any doubts or any clarifications, please feel free to ask.
Hello, sir.
Hello?
Good morning, sir.
Good morning.
Sir, it's a very nice presentation.
Thank you.
You have given the insights of the pharmaceutical research in terms of phytopharmaceuticals. Sir, I would like to know, when you have done the HPLC of a neem sample, have you used the same mobile phase for identification of all 4 different phytoconstituents, or you have used different mobile phases?
HPLC? No, before finalizing any HPLC method, first we have to do the development of activity. We have to run different solvent system. Okay, different solvent system.
Yes, sir.
And then different nanometer you have to identify, because I told you,
Yeah.
You have to identify 200 nanometer and 325 nanometer, 370 nanometer, 254 nanometer, so that we can see that how many bands we can see in a significant way. And then we can correlate with the literature available. What are the different, as per the literature, what are the different molecules, what is the structure of the molecules, and what is the UV lambda max of those molecules. Based on that, we can see that, and we can select the wavelength, and we can identify the molecules. First, before you going for any finalization of the method, first you have to develop in a different solvent system and do that development study and finalize. Of course, this is the only one method, but in different nanometers.
Yeah, but sir, have you used the same mobile phase in all four identification?
Yeah, the same mobile phase.
Sir, but how it is possible, sir? The same compound at 200 nanometer may show some other peak at 355 nanometer also. There may be a chances of the showing the same peak.
Because see, when developing, we have to make sure that, because see, overlapping in different nanometer is not possible, because whatever the lambda max is having, whatever the molecule is having the lambda max, only those molecules are identified in the chromatogram. Other molecules we cannot identify. Okay, because the phenolic acid, suppose phenolic acids is the lambda max is around 325. Only phenolic acids are available. I can see that phenolic acids in the significant way in that particular chromatogram. If you do 200 nanometers, other molecules, either triterpenoids, any terpenite molecules will be showing the lambda max at 200 nanometer or 210 nanometer. But 254 nanometer will not interfere in the 200 nanometers. 325 nanometers compound, lambda mass compound will not interfere in the 200 nanometers. All compounds is having their own lambda max. Based on their lambda max, you have to choose the nanometer.
No, sir. As per my knowledge, there is some or the other, the particularly the interactions, or there may be some absorption of the lambda max will be there about this one.
Yeah.
You cannot get directly only the particular filter.
No. Yeah, presently using this latest technology, we can go for the gradient, and we can do the permutation and combinations, and
yes,
even permutation.
Yes, if it is a gradient, there is a chances.
Yeah,
yes, if it is a gradient illusion, yes, there is a chances of identifying a different phytoconstant at different lambda maxes and different position. But with the same isocratic gradient, I don't think so, sir.
No. Isocratic it is not possible. It is not possible.
Okay, thank you, sir.
Gradient, different nanometer you have to use it.
Hello.
Hello.
Sir, this is Dr. Basraj from SGMCP Chitraga, sir.
Oh, tell Basraj, how are you?
Fine, sir. Okay, here with the small doubt I am asking. So, microwave radiation and extraction of phytochemicals. Regarding this, what about your opinion, sir? So many papers are there. What about your opinion on this regard?
Microwave radiations?
From that, using the microwave extraction of phytochemicals.
In the phytophobicals, whatever the technology you are wanted to use it, it is all possible. Whatever the technology you wanted to use it, you can do it. Only thing is, for each and everything, there is a guidelines. Okay, when you are wanted to register any product or sell any product for any country, each country is having their own guidelines. Even whatever solvent you are using, residual solvent, you have to meet as per the AICTE guidelines. If you are using as a microwave race, for that each and every country is having the guidelines. Should be meet that requirement. If it is meeting that requirement, then fine, we can register your product.
Okay, sir. Sir, one more thing. In academics, whatever we are giving extraction for students, so we are facing some problems with the solvent, sir. Maybe band separation will be difficult, and isolation. We are facing a problem with isolation, sir. What you can guide us regarding the selection of solvents, sir? whether which company is preferred or what kind we have to make, how we have to select the solvent. We know the idea what solvent we have to take. But thing is, so even though the company will send some solvents, they are not being good standard. So in this regard, what you are going to say, sir?
See, first question is selection of the solvent. For selection of the solvent, based on the polarity, we all know that based on the polarity, we have to select the solvent. Okay, suppose if you are using that methanol extract, whether which extract you are using, whether you are using a methanol extract or hydroalcoholic extract or chloroform extract or ethyl acetate extract, based on the what extract or fraction we are doing the chromatography, you have to understand. Suppose if you are using that chloroform extract, ethyl acetate extract or hexane extract, always these are the non-polar solvent you have to use for the non-polar solvent combination. Suppose if you are methanol extract or ethanol extract or hydroalcoholic extract, you have to go for the polar combination of solvent, either butanol acetic acid water or chloroform methanol water, or methanol and water based combination. You have to go for the methanol and ethanol and water extracts. And if it is chloroform ethyl acetate and diethyl ether hexane, only chloroform ethyl acetate, chloroform diethyl ether ethyl acetate, chloroform or toluene ethyl acetate. For non-polar extracts and fraction, you have to go for non-polar combination. For polar extracts, you have to go for polar combination. That this is applicable for the TLC. If you are going for the column chromatography, again there also what extracts you are loading for the column chromatography, but you have to start with the non-polar solvent. Suppose if you are loading that ethyl acetate fraction, you have to start with the hexane. You have to start with the hexane, followed by the chloroform, followed by the ethyl acetate with some percentage addition, increasing the some potential addition. Suppose if you are going for the methanol fractionation, directly you can load the sample with the chloroform with increase of the polarity of ethyl acetate, followed by the some percentage of methanol, followed by 100% methanol. That way we can do in the column chromatography. And finally, it is a trial and error. Based on the experience, we will get the idea.
Thank you, sir. Thank you. Thank you for your valuable suggestion. And actually,
Thank you.
We are faced the problem with the whatever the reagents we are going to use for this. When you are placing these solvents on column, we have to go for the purification. What we are noting, because I don't know, the standards of chemicals is going to be varies. Actually, we will fail in that. In first step, okay, second step, okay. So if you add any other solvents, so where the separation will be difficult, and we have faced some problems while separation, sir.
Main thing is main problem we are generally facing, because everything they are looking in the only one column chromatography, purifications. Purification is by single chromatography is very, very difficult. We have to go for the multiple column. Suppose if you are doing any column chromatography, first you have to do that crude fractionation. Then you have to give that numbers, different fractions, and you have to identify by TLC or by HCLC, and you have to identify which molecule or which peak you are looking, which band you are looking. Based on that particular fractions, you have to combine it, and you have to go for further fractionation by multiple columns. Okay, and meantime you have to note down in which fraction, which polarity your molecule of interest is eluted, and you have to, based on that polarity, you have to go for next further fractionation by column chromatography. Repeated column chromatography, definitely we can purify to even more than 90% percentage of molecule.
Okay, sir. Before going to column separation, is it going for HPTLC and it is going to give a good information which solvent you have to select
Yeah,
and which.
definitely.
Okay, sir.
Yeah, suppose if you are going for the, before you are going, don't go directly for the column chromatography. First, you have to identify by the HPTLC or HPLC, by TLC, which, because based on, you will be having the literature and which molecule you are looking at. Suppose if already literature is available, which molecule you are looking, which RF value you are calling molecule of interest, it is already in the TLC. That time you can identify your molecule either by UV or fluorescent or spraying. Then you identify this is the molecule I am looking, this is the RF I am looking, and based on that you have to isolate this molecule. First you have to identify by TLC. Then based on that you have to go for the column chromatography. Then all the different fractions you have further you have to do that TLC. Then in which fractions your molecule of interest is present, then you have to combine those molecules and further you have to rerun in the column chromatography. In the same way, you have to do multiple column chromatography until you get that purified fraction. That purified fraction, the purity we can establish by either by HPLC in different nanometers.
Thank you, sir. Thank you.
Thank you.
Sir, I have one more question. Sir, this is Anand Bhandarkar from Goa College of Pharmacy.
Okay, tell another one. Go ahead.
Sir, have you done any effort for the masking the bad smell of neem oil?
Neem oil. See,
Yeah,
this
seed
is
oil.
the neem oil. Neem oil contains so much of phytomolecules that
Yes, sir.
contains so much of bitters. Okay, heavy bitters contains, and these almost all the bitters having the pungent in nature, and
Yes,
these
sir.
are the bad smell, the inherent character of the neem. Okay, if it is not, if a bad smell is not from the neem, that means it is not a neem. This is the inherent characters, because since it contains all the bitter principles.
Yes, sir.
This bitter principles also is the active principles.
Yeah.
Yes, good morning, sir. Myself, Ajit Patil from ASPM College of Pharmacy. I have one question regarding the extraction techniques, sir. The newer technique that is phytonics is there, sir. So I want to know about the technique which is applicable at institute level. How we can implement that technique at institute level. If I want to give the demonstration of any constituent extraction by the phytonics, then what is the possible method or any technique is available in India to show the students how to utilize the phytonics?
Phytonics. Regarding phytonics.
If any information you have regarding the phytonics techniques, then you can share afterwards, sir, also. Okay?
Okay, thank you.
Okay, sir.
Okay, thank you, everybody.
Thank you so much, sir.
Thank you, sir. Thank you very much.
Thank you.
Okay.
Thank you, Dr. Prakash, for giving valuable suggestion as well as information. And I think all the participants, they have enjoyed sir's lecture because it is an interactive session and all of you clarify your doubts.
Okay, thank you. Thank you.
We are very much thankful to Dr. Prakash for sparing his valuable time. Even though he is busy, he has spared some time for a QIP program. Even he is the instrumental person to arrange our next speaker, Mr. Rajendra. So he is his colleague. And now we will continue with Mr. Rajendra sir. So I request Dr. D.N. Sastry sir to introduce our next speaker, Rajendra.
Okay, thank you. I have a very pleasant task in introducing Rajendra Bojnala. a very skilled professional with having more than 17 years of analytical research and development experience in APIs and ANDA formulation. Currently, he is working as a senior research scientist, R&D at Himalaya Drug Company, and he has a very rich experience at Campbell Biopharm Private Limited as in R&D as a senior research associate. As well, he has a very good experience at AICTE Pharma Development Center and at Cadila Pharmaceuticals, Syngene International Limited as a research scientist. With this, obviously it is very clear that he has got a very rich industrial experience, and he has also has a very good hands-on experience on various advanced instruments such as HPLC, LCMS, GC, and preparative HPLC. There are so many lists of the instruments which was provided in the CV that will give us motivation that I'm sure that the lecture will be very interesting. And he has, in addition, he has won a Best Paper Award in an international symposium conducted at Shivaji University in the topic New Drug Discovery and Process Research. Also, he won the Scientist of the Year Award at the Himalaya Drug Company for the year 2015 and 16. And he has got a very good, he's undergone a good training programs in fundamentals in LC-MS analysis, as well as applications of the NMR for biopharmaceuticals and protein analysis. He has got he's pursuing currently his PhD from SRM University and has got a master's degree in quality management from SMU Manipal, and his master's degree in pharmaceutical chemistry from Alamein College of Pharmacy and a bachelor's degree from KV College of Pharmacy. With this brief introduction, on behalf of the Kelly College of Pharmacy, on behalf of the organizing committee for this quality improvement program, I wholeheartedly welcome Rajendra Bhojanala for his lecture.
Thank you very much, Sir Principal Sir, respected Principal Sir, and all the respected faculty members who are attending the program today. And it's a great honor for me to give a presentation to such highly qualified people and experienced people. So I would like to just share some of the experiences and observations in my professional career till now in the 17 years of pharmacy career. So I hope you will definitely like the presentation, and maybe you can make it a better interactive session if there are any doubts later. So with this one, I will just start my presentation. Yeah. Good morning, one and all. So today I am going to present my topic as advances in phytopharmaceutical chemistry and in synchronous with the regulatory perspective. In synchronous with regulatory perspective, and I am working in Himalaya from past 7 years, and with my experience of phytopharmaceuticals and regulatory aspects, in comparison with APIs or any synthetic chemistry molecules, is a very big challenge for me to understand, and I have made my best to come up with this kind of presentation so that you will also be able to analyze and get updated with the current scenario. So yeah, as my colleague Dr. Prakash has clearly given you an idea about the present scenario of herbal drugs and the growing popularity of the herbal drugs in the industry, which has come from a basic of 62 billion in 2005, and now it is popping up like anything, and in 2020 we can see it has crossed more than $5 trillion business all over the globe. And now we are well aware of the craze which is coming up for the herbal drugs because of its lower side effects or minimal side effects or for the better potentiality or dependency of on the medications for longer durations for many of the ailments, which has to be taken for more than 3 months, 6 months, or 1 year courses. So synthetic drugs are not reliable, and because of their other side effects, which are mainly a reason for them to get a drawback in the present scenario, and because of herbals which are having no side effects or more technology is getting put into these herbals nowadays, these herbals are picking up like anything. And this is only a small figure of 5 trillion, and it can go much more. And in this COVID scenario, you have whatever heard, only 2 or 3 drugs of APIs have come into the picture as in vaccines, but a lot of herbal drugs have been coming to action, and a lot of it is like a mouth-to-mouth spread of drugs is happening. So because of this mild side effects and more curative actions, and for longer durations, these herbal drugs are picking up like anything. So coming up, how these herbal drugs are taken or how these herbal drugs are used, these are divided into categories of traditional medicines. So as per our age-old culture, like Charaka Samhita or Sushruta, who has given us a beautiful knowledge of these herbal drugs. So we are using these as a household medicine also. So we call these as traditional medicines. And the patent or proprietary medicines based on the recent technologies and updations, whatever is happening for individual markers or specific markers or some group of or polyherbal categories instead of one single medicine. These patent and proprietary medicines are one category. And coming up these as in dietary supplements or food nutrients or replacement of the existing food for some of the people who are very much of malnutrition or undernutrition. We can use it as a nutraceutical or health supplements. And nowadays, based on the aestheticity and the modern tradition which is coming up, nowadays people are more focusing on the aesthetic properties or fashionable properties in respect to these cosmetic products. And phytopharmaceuticals, as I told you, is very much picking up with this respect to ethnobotany, or because of these advancements in herbals, phytopharmaceuticals is becoming equivalent to that active pharmaceutical ingredients, wherein we are targeting certain molecules for some particular clinical properties and enhancing the clinical efficacy of the molecules, which is called as phytopharmaceuticals. I think my colleague Prakash has clearly stated some of the importances of phytopharmaceuticals in his previous, in his earlier presentation. So why this requirement of advancements or phytopharmaceuticals or herbal medicines is required means, from the, if we see the shift from the individual medication which was taken as a household medication, or some rishis or munis would have given this individual medication, but now based on the requirements and the benefits which are coming from the herbal materials or herbal formulations, there is a drastic shift from the individual medication to the large scale production, which is the present day requirement. And to meet this supply and demand, there are a lot of stringent conditions or stringent requirements or regulations which are coming up in respect to this safety and efficacy of the phytopharmaceuticals. So there is a requirement of a lot of advancement and technology to be put in now to see whether these medications are safe and efficacious. So what are these herbal materials we analyzed for? So one main reason for the analysis of these herbal materials in comparison to the synthetic molecules, if you say, the standardization of the materials which is consumed in the product is very much important. Because we are synthesizing a molecule and we are seeing whether there are any residual impurities or some related impurities in the molecule. But in herbal material, it is not like a single molecule or a simple material which is available. So there are a lot of variations which come into picture because of climate, because of cultivation, because of soil, and many other variations put into this. So a standardization procedure of these molecules is very much required. And as you know, these natural materials are always available in different species, different conditions, and other different environments. A lot of chances are there for adulteration of these materials. So it is always an important thing to identify the main prime material and adulterated material. So our concept of safety come into picture here. So adulteration is very much important, and we have to analyze for adulterations. And as I told you, safety concerns are more important, as now we are for commercial purpose or for profitable purposes or non-availability or lack of knowledge, people can consume a wrong thing, which can be a safety concern over here. So we have to identify, standardize, and analyze the material for their toxic compounds or adulterated compounds, which can cause many of the side effects. So safety concern is one of the important things for which we have to analyze the herbal materials. And because of this growing popularity, now too many companies or too many people are into this, focusing on these herbal medicines as one of the important criteria. We are all having a lot of knowledge on this one. As I told you, this is not only a traditional medicine, this is a household medication. So in our kitchen is like a regular herbal medical shop, you can say. We have a lot of medicines available in our kitchen itself. So we ourselves are doctors in our kitchen. So, but this is not applicable to each and every country based on their global requirements and based on their conditions. We have to meet their regulatory criteria, where some things is available in India and something is not available in India, or something is not acceptable out of India. Like for example, when we use berberine or solosodine, we Indians are very much useful, very much using this bale as our one of the important food material. But this bale is not acceptable when berberine or solosodine is there in Singapore and Malaysia, because of their genetic nature and culture over there. So when we have to market a product which is having Hegel marmalose or bale, which is having solosodine and berberine, it is not acceptable in Singapore. It should be free of these both materials in Singapore to get a product to be registered over there. So there are like this. This is one example. So these kind of examples are available plenty, and a lot of governments have their own regulations, rules and regulations to meet their criteria and products to be registered in their countries. So we have to analyze and keep the data of all these materials when we have to go for a different regulatory environment. So what are the typical properties which have to be analyzed? As I told you in the earlier slides, so based on the chemical composition of the molecule, we have to have all the data, whether it is of safety concern or if it is of a efficacy concern. So the chemical composition of the raw material or the finished product is very much important, which has to be kept on top priority when we are using it as a medicine. And the physical properties, this depends for branding of the product or imaging of the product to improve the aestheticity or palatability of the product. So these physical properties have to be considered. And for checking these adulterations and toxin levels and all, these microscopical products are very much important for us to standardize the materials also. And sensorial point of view, that is, as I told you, like palatability and the view, what do you say, the patient should invite that product, means that he should be able to easily take that product by seeing the product itself. So to improve all those sensorial characteristics and all, we have to be analyzing the products. So when we have so much to analyze and we have so much to standardize, so what are the challenges which are which we regularly face during our product development and marketing stage? Means there are different stages for these challenges, like development, manufacturing, marketing, and profitability. Each and every stage we face a challenge. So coming to the development section, when we say there are a lot of high development costs involved and the clinical complexities, because these medicines, traditional medicines or whatever the herbal medicines are there, we have a limited knowledge of these compounds. Even when we consider about the Rishi Muni Granthas like Charaka Samhita and all, and we're now coming to this Ayurvedic pharmacopoeia of India and all, we have a limited amount of knowledge over there. So when we are targeting for a particular system or when we are using it for an advanced requirement, these available trend data and all is very limited. So we have to develop our own data in our based on our present requirements, and we have to get on with the clinical complexities also because of these new requirements, because we don't know whether these are analyzed, whether the data is collected in respect to these clinical challenges or not. So there is a lot of challenges involved in this developmental and collecting the data and establishing the clinical efficacy of the drug. And coming to manufacturing, we don't know how the materials and all behave when we are going for using a different compressing machines or blending machines or whatever for used for washing and the filling machines and all. So all these things can be like, because of their unknown natures, can cause a lot of technical barriers, and manufacturing facilities have to be updated based on the requirements. And coming to the marketing, as I told you, different countries have their different regulatory environments and guidelines. So to meet these regulatory requirements without having prior knowledge is a big challenge over there, and we have to establish plenty of data regarding this one based on the available data, based on the collection of data and establishing the data. And when we come to this profitability, as we know, a lot of people don't know the beneficiary, might be not aware of the beneficiary of this herbal drugs, and also for promotion and brand imaging, we have to spend a lot of money on these products for either advertisements or for making the doctors understand about the efficacy of these drugs and clinical safety of these drugs. And we have to create a lot of data also for establishing, so that also add up to the cost of the marketing and profitability margins. And as I told you, we have different regulatory environments, and when we come to the domestic market or Indian market, we have a very limited, or like there are still progression is going on, or work is going on in imposing the regulatory guidelines and requirements in India. And presently the safest one is I have mentioned here, like AYUSH, FSSAI and CDSCO, where AYUSH looks about the herbal medicinal products with respect to Ayurvedic, Siddha and Unani, and FSSAI is more related to the dietary supplements or food or nutraceutical products, and CDSCO is more related to the cosmetic products and the phytopharmaceutical products. So we are seeing only three regulatory major regulatory bodies are there in India to guide the guidelines. But when we see global market, we have a lot of guidelines which are guidelines and regulatory requirements which are guiding a molecule to be registered or consumed in their countries, like ISO, FSSAI, AOAC, FDA, Codex Alimentarius, Food Safety and Standards Authority of India, that is European Commission is there. So these are least Sante guidelines are there. So these are the few guidelines which commend a medication to be registered in their product, and they have their own safety and clinical data to be established, toxicology data to be established for a product to be registered in their country, because this approach is called as a farm to fork approach, because we don't know what is getting grown in agricultural land, how it is getting collected, how it is getting processed, how it is getting filled up, and how it is getting labeled, what and all is getting labeled on that one. So unless until a man consumes, that is on his plate or in his hand, they are having a lot of guidelines to regulate the market. So few of the challenges what we face in terms of research, when it comes to research, is we don't have any perfect sampling procedures as with respect to an API pharmaceutical marketing, like active pharmaceutical markets. Like we have different formulas like root N plus 1 or 6N system or 9-point system or 12-point system we have for sampling. Their uniformity of the sampling will not be such a big issue, but in terms of herbal medicines, we use a lot of heterogeneous materials like roots we use, stems we use, buds we use, fruits we use, seeds. So like these kind of lot of different variant materials we use. So heterogeneity which is available in the Ayurvedic medications or herbal medications will pose a major challenge in sampling the large size samples. So the sampling procedures have to be adequate and establish a good sampling procedure for minimizing the sample uncertainty in the analysis. Second one, the method of analysis and the measurement uncertainty is a major challenge in herbal medication. It is not like in a simple API molecule, because we don't know first of all what is there in the molecule. And in our colleagues earlier session, one doctor asked about a question like one single mobile phase is sufficient. Yes, we have a lot of research to be done on those kind of molecules and procedures, because maybe we are lagging behind. We are seeing into one angle, but when we use a different mobile phase and different technology, maybe few other compounds can get popped up. So what method of analysis we are using is sensitive enough or not for identifying that itself will have a major challenge and time consuming process over there, and a lot of studies have to be taken up over there. And measurement uncertainty, that depends on the technology what we are using, the amount of material what is available in the molecule, in the compound. So it always poses a challenge in the reproducibility of the analysis what you have performed. Third one is mixture of these things, that is screening approach, which is a sampling and analysis together combination. So if you are having a large amount of sample and a hi-fi technology where you have to inject only a small amount of sample, again there is a mismatch. Or if you are having a very less amount of substance and a very low sensitive instrument, it is a mismatch. So there should always be a screening approach for sampling and analysis, and both should be correlated. Only major thing or goal in our required in our research or analysis is we have to achieve ultimately a very low rate of false negative results and or acceptable rate of false positive results. That is our aim in the analysis and research. Coming to the real-time challenges of analysis, what we exactly face when we come onto the bench or when we use when we put our hands on our instrument is the one of the thing is complexity of the matrices. When comparison of APIs or single synthetic molecules, one major issue is the complexity of the matrix. We don't know what and all is there in the herbal material. It can be a mixture of all the things like proteins, carbohydrates, fats, oils, fatty oils might be there, essential oils might be there, and a mixture of other starches, routines, everything might be, gelatins, everything might be there in that. But we don't know exactly what is what it contains. So we cannot simplify a material. We cannot just simply make a material into a simple form and try to inject or analyze it in a easier way. So this complexity of the matrix is one of the major challenge which has to be considered, and we have to be a target oriented approach should be there for analyzing these kind of complex matrices. So exactly what you want to analyze proteins, analyze it for proteins. So we have to separate all the other materials from the proteins. That should be our approach. So this complexity of the matrix is one of the important thing which is a major challenge in the analysis. Second one is diversity of analyzed tested. As I told, the heterogeneity which is there in the herbal molecules. Like if you are using seeds, you might end up with a different pattern of molecules or profiling of molecules what you have done. And if you use a root of the same plant, you might end up with a different profile of molecules when you analyze it. So the diversity of the analytes which are present in different heterogeneous parts of the plant or the herbal material is a major challenge, and target-oriented approach is required over here also. And one more major challenge is this ever-growing list of target analytes. As we know, earlier we had only a few analytes which were isolated or characterized and published, but now, based on the available technology and the advancements in the technologies, there are always a growing list. Today, you have 4 analytes in a molecule, in a compound. Tomorrow, if somebody uses some more better technology or sensitive technology, maybe you will end up with 8 molecules over there. So, the ever-growing list of unknown molecules which is added into the list is a major challenge. So, it is a major challenge in the analysis. And this default MRLs, that is minimum residual levels, are very low. For these levels, because of the availability of the analyte, major analyte or targeted analyte in a herbal molecule itself will be in maximum like ppm range or ppb range. So the availability of the material itself is a big challenge, and isolated material is another challenge. So this default MRL values is a major challenge in the real-time analysis. And knowing the unknowns, as Dr. Prakash told, in neem we have characterized 4 molecules, but neem doesn't have only 4 molecules. If you see, there are a lot of other terpenoids and flavonoids associated with that in the same HPLC chromatogram. So we don't know what and all is there additional to that 4 main major peaks in that one. So maybe that is one of the important thing. what we have to analyze means it takes a lot of efforts and challenges to overcome those things. So after seeing all these challenges which are posed in the herbal materials, the advancement is always an answer for those things. And one answer for that is hyphenation technology of instrumentations. This hyphenated techniques ranges from the combination of simplified separation technique, or a separation identification technique, or an identification technique. So here I have just given in a brief skeletal diagram where a simple where an sample processing technique like SP, that is solid phase extraction, or large volume injection can be taken, and it can be used like simple instruments, GC, HPLC, or column or capillary electrophoresis, and it can be combined with the detection techniques of PDA, IR, or MS, or NMR, or TLC. And further, for this is the basic instrumentation, and further it can be combined into a different options of GC, PDA, GC IR, GCMS, GC NMR, or GC TLC, or HPLC IR, HPLC NMR, like this. Different instruments which are used for separation and detection can be put into hyphenation and made into a better sensitive and advanced technique. So this hyphenation is the answer for all those requirements of the regulatory or for the challenges which are posed on the table with respect to herbal materials. So coming to the advantages of hyphenated techniques, these hyphenated techniques are always fast and accurate results can be obtained from this technique. They give a higher degree of automation, so that as I told you, all the three combinations can be used, or a simple combination can be used for our purpose. And they give the sample throughput will be high. So earlier it was a very big challenge for us to analyze the samples, and time consumed was more. But nowadays this automated technology has come into picture, and because of this hyphenated technique, the sample throughput is always more than the older periods. And better reproducibility because of the higher automation which is obtained, and reduction of contamination due to its closed system, because earlier we were using in one system, collecting a sample, injecting into another system, getting the results from there, and passing it to some other system. It was all like involved into too many steps were involved in that one. But now it is not like that. Just keep the raw material here on one side, connect the techniques of hyphenated different instruments in a series, and get the complete results of your choice. So you are not taking the compounds or putting hands, or no contaminations or any other problems, hindrances are there. So it is a very closed system, and other interferences or contaminations can always be avoided. And lastly is that separation and quantification is achieved at the same time. As I told you, you don't have to again and again put hands onto the sample and load it into different systems. Now a lot of things can be interconnected, and a simple separation and the advanced quantification, everything can be achieved at the same time. Here I have given a few list of hyphenated techniques like GCMS, LCMS, LCNMR, ECMS, GCIR, LCMSMS, GCMSMS, GCMS, GCNMR, and GCAS. So with this list of hyphenated techniques, we can cover all this separation identification, and quantification all simultaneously. So when we apply this hyphenated or advanced technology to a simple traditional method of TLC, that is thin layer chromatography, we can see what wonders it can do. So here I have just given a skeletal diagram, or a simplified HPLC system which is hyphenated. In this one, we have auto sampler, we have chromatogram development chamber, which is a derivatization chamber, a detector or a densitometer, and a documentation chamber where we can have different wavelengths of 254, 366, and white light. And finally, it is connected to an this is the one which is connected to a QDA, that is a single quad mass spectrometer. So here we can see all these things are interconnected. Here I have not shown the connections, but these are all interconnected. So when you just prepare a sample, you can just place it over here. So the sample gets automatically applied here, automatically developed in this plate, automatically derivatized. We are having a densitometer which gives all the peak separations, and the quantification is possible in this one. We can see the separations and recordings of all the chromatographic different bands, and different derivatization procedures give you different compound details also. And finally, if you are interested in any particular sample, it can be directly connected to an interface where the sample is, where is a spot, that particular spot is getting analyzed, and we can get the mass details synchronously in the single quad detector. So only by injecting a sample, you will be getting all the details, and even characterization is also possible to an extent with this hyphenation technology for a simple HPTLC system. Here I have just given a brief about a simple instrument hyphenation technique, what I showed in that earlier slide. So here we have connected a LC system to an interface of TLC and a single quad detector. Here the HPLC pump is used at a 500 microliter per minute capacity, and a CAMAC TLC MS interface 2 system is used. Here the spots which are developed in the TLC plate are placed under this TLC MS interface, and a QDA detector is connected, which is an electron spray ionization technology one. Here this is an elaborated, means like enlarged image of the TLC interface, where we can see here this is the plate which is placed under the TLC MS interface system, and the positioning is done in such a way that the laser which is there inside that CAMAC TLC MS system is pointed to the spot of the required, is pointed towards the required spot which has to be analyzed in the MS detector. So here we can see there are 3 different colors over here, and blue color is there, and here this is enlarged image over here. The blue spot, whatever is there here, and after applying that 500 microliter per minute mobile phase from LC system, this spot gets dissolved and absorbed into the QDA detector system, and directly we give it gives the mass of the required mass of that absorbed spot from the TLC plate. So out of these spots, we have just selected one blue spot, and we have characterized or identified the molecular mass of that product. So we can now use this hyphenated technology from simple separation to identification and characterization also. And coming to these basic instrumentations and basic instruments what we are using regularly, like HPLC, there is not much of advancements in the HPLC except that high performance liquid chromatography, which is a fast injector, and high pressure tolerance will be there, so that we can use a columns of different dimensions and a lower particle size columns also. But there are a lot of advancements in respect to HPLC detectors what we use when compared to earlier UV detector or photodiode array detector. Now presently we have fluorescence detector, refractive index detectors, evaporative light scattering detector, charged aerosol detector, electrochemical detector, and mass spectrometer. Out of all these HPLC detectors, in particularly ELSD, charged aerosol, that is CAD, and mass spectro detector are the one which is having much more advancements going on right now in the current scenario also, and they are evolving like anything day by day. So today the detector model whatever is available with respect to this MSD or ELSD is not available tomorrow, or it is much more advanced tomorrow to answer much more queries also. So there are a lot of advancement happening in respect to HPLC detectors. And the same advancements is happening in different column chemistries also, like compared to these earlier normal phase columns and rapid reverse phase columns. There are a lot of bonding which is happening here. Presently, the trend or scenario is with respect to Hilli columns. That is called as this hydrophilic lipophilic interaction columns, wherein we can convert these columns into normal phase as well as reverse phase also. And based on some chemistry tweaking and availability, we can convert these columns into highly polar columns or highly non-polar columns. So the versatility which is presented with these Hilli columns is very much useful for a molecule to be having a good profiling system by which to analyze a molecular profile using these kind of columns. And next technology, which means like next further improvement in these columns is, as I told you, that is ultra high performance liquid chromatography columns, wherein we see lesser particle size of the columns. Earlier we were using around 3 micron columns was the maximum one available. Now we are having 2.4 microns, 2.1 microns and 1.8 microns columns also are available, which gives much more resolution and theoretical plates. Much more column efficiency will be there in your for better profiling of the molecule. And this 1.8 nanometer 1.8 micron column is going to be it's almost like 2 or 3 molecules are already published in United States Pharmacopoeia, and these are nowadays getting applicable with respect to this QBD, quality by design approach, is having a lot of applications with this UPLC columns. And as I told you, there are a lot of unknown molecules or lot of unknowns which are getting presented day by day. And with respect to these regulatory guidelines, they are becoming very much stringent in respect to these unknown molecules or newly known molecules which pose a danger for the health and safety of the consuming person, or with respect to human beings or animals, because these are given as medication in small amounts and dietary supplements in large amounts, and that too for larger periods. So the newer analytes or toxin analytes which are popping up every day in our herbal materials are major concern. One of the higher priority concern is these mycotoxins. These mycotoxins are naturally occurring toxins produced by certain molds, that is fungi of Aspergillus species, Penicillium species or Alternaria species, and these are regularly found in the food materials. This can be present in the food materials what we are using as a dietary supplement, or it can get cross-contaminated because of the co-cultivations and other scenarios, or the harvesting seasons and the storing conditions and all. And these mycotoxins have a variety of adverse effects and pose a serious health threat to both humans and animals both. So the adverse effects of mycotoxins ranges from small acute poisoning to long-term effects such as immune deficiency and cancer problems. So these are of high concern right now from the regulatory markets. So when we see these regulatory requirements of mycotoxins, I have given an example of this Latin America countries regulatory requirement. Here we can see Argentina, Brazil, Mexico, Chile, Colombia, Trinidad, Jamaica, and Suriname. They have different requirements of the levels levels of mycotoxins to be present in their compounds. When it comes to aflatoxins, Brazil has a very stringent limit of 10 ppb when it comes to human consumption, and 10 ppb when it comes to aquaculture in Colombia. So we have a largest or stringent limit from Brazil only. Everywhere we can see that is a stringent limit which is posed, but very relaxed limit is Argentina, where we can see a lot of scope is there for presence of these mycotoxins. And critical mycotoxins which are analyzed are aflatoxins, ochratoxin, zearalenone, fumonisin, deoxyvalinol, T2 toxins, and diacetyl So even milk what we consume is also having mycotoxins in the form of M1. M1 and M2, majorly which is M1. And coming to one of the other impurities or expected impurities is residual solvents. This is a general impurity which we can expect because of our processes which are involved in the development or standardization or production or packaging of the formulation herbal materials, because we use a lot of solvents to either extract or purify or clean or solubilize. So we use a lot of solvents over here in the manufacturing process, during manufacturing processes and after the product formulation. So residual solvents are defined as organic solvents or volatile chemicals that are used or produced in the manufacture of the drug substance or excipients or in the preparation of the drug substance. So each and every country has a different requirement, and ICH has given a different classification of these solvents and based on their toxicological data or LD50 data. And they have classified these residual solvents as class 1, class 2, and class 3 solvents based on their carcinogenic effects. Class 1 are the solvents which has to be avoided in major cases, and their presence has always to be justified. Class 2 residual solvents are some of the solvents which we use it for our regular routine purposes of manufacturing or extraction, but they are allowed at a certain ranges, and the toxicology and clinical data is established for the presence of these solvents. And class 3 solvents are the solvents which have minimal toxicological data, and it can either be rejected or approved based on the justification and the amount of solvent which is present in the molecules. So for establishing a good residual solvent profile, or to get a good results from the residual solvent for the residual solvents, the hyphenated technology what we use is a simple GC system hyphenated with an Headspace analyzer. So we have this is the diagram which is shown for GC, simple GC, which is connected with an Headspace analyzer. And this Headspace analyzer always have the edge of analysis or the sensitivity when compared to direct injection, where we use a direct solvent of 1 microliter or 2 microliters for an analysis. But this Headspace analyzer will concentrate the sample in its Headspace. The space what is available above the solvent or the sample level is called as the Headspace. So because of the thermal heating or thermostatic heating, we are creating that vapor from that solvent whatever is there, and we are injecting the vapor within a highly concentrated form into the GC. So this hyphenated technology of Headspace is always having more sensitivity and more concentration of the minor constituents which we are looking out for analysis. Like these mycotoxins and all are present in ppb level, so directly sampling technique to get the ppb and molecules will be very difficult. So we have to concentrate that molecule before injecting, and this Headspace always helps in the process. And these Headspace are available in different forms. Like this is the static Headspace sampling unit what I have shown here, where it is a simple manual gas-state syringe or automated Headspace analyzer needle is injected into the thermostated oven, and the direct vapor is collected into this manual gas-state syringe and injected into the GC. Second one is the dynamic Headspace trapping device, which is here we can see there is an inert gas which is passed into this Headspace, and the adsorbent or cold trap assembly is attached to this one, and directly the ingredients are adsorbed into this adsorbent when an inert gas is passed. So here already this gas is getting concentrated analyte percentage, which is further more concentrated and free of interference. It's in this adsorbent cold trap. So static Headspace is a general analyzer, and dynamic Headspace is much more advanced technique than the static analyzer. But present day scenario, we have much more advanced technique called as Persian trapping technique. Here it is like the same dynamic Headspace syringe technology only, but here the adsorbent or cold trap which is there is still concentrated by trapping continuously the inert gases which are passing the analyte into this adsorbent for 2 or 3 times or whatever the requirement, or based on the lower level of the analyte what you are analyzing. It further traps that analyte here, and once completely there is a bypass over here. This it's not showing. Okay, there is a bypass assembly will be here. So directly inert gas will be purging that adsorbed analytes of lower concentration at a very high speed into the GC, where you can get a more better sensitive peak of the lower analyte which is present. One more major challenge in our herbal products is these pesticides, as they are used for many purposes. They can be used for the different utilizations, like they can be used as insecticides, herbicides, rodenticides, and other purposes. And based on the chemical identity, we can identify them as organophosphorus compounds, organochlorine compounds, bromine compounds, and acidates compounds. Like this, there are different chemical natures of these pesticides what we use. And the regulatory guidelines, right now it is still in a dilemma to phrase a single or global harmonized rule for these pesticides, because whatever is approved in India is not at all available in US market or America or European market. And whatever European market is available, India has completely a ban on those usage of pesticides at all. So there is nothing like and there is no like harmonized rule that these cultivation should be sprayed with these pesticides, and these are the only contaminants of pesticides which will be available for a particular cultivation or a crop. So there is right now it is a big dilemma in global scenario also for harmonization of these pesticides. So based on the regulatory requirements and other necessary conditions, we have to analyze these pesticides in a much more sensitive way. When I give an example of European production, European regulations, and as per the Environmental Protection Agency guidelines, they have classified these pesticides also into 4 classes, that is 1, 2, 3, 4, based on the toxicity level of the toxicity threat to the health. If it is claimed as class 1 toxic, then it is very highly toxic and it should be compulsorily labeled as danger or poison. When medication is having class 2 toxicity, it can be given as it can be termed as moderately toxic, and there should be a warning symbol compulsorily on the label. And the class 3 and class 4 are slightly toxic or practically non-toxic, or still toxicity data has to be established for that one. There we can give a precautionary notification as caution when we are consuming those products. So establishing the level of pesticides or the amount of pesticides or the kind of pesticides which are present in the herbal materials is a very important task, and this GC-HS technology or GC combined with MS technology give us that edge on establishing the profiling of pesticides. One more major threat to the herbal materials is these dioxin compounds, which are which can be a synthetic or a semi-synthetic contamination also, and these dioxins are very much carcinogenic when it is consumed in a higher quantity. So these dioxins also to be analyzed in the sensitive technique by hyphenating GC MS. Recent advances and regulatory requirements with respect to herbal materials is this GMO, what we call it as genetically modified organisms, whose genetic material has been altered using genetic engineering techniques. One example I have given here is the 35S promoter and NOS terminator, where this 35S promoter is used to increase a certain genes in a molecule and to give an enriched content of that material. And this terminator is used to decrease a toxin level or non-approved or not required materials in a plant or organism. But both the approaches have their own pros and cons, so we have to be cautious about the effect which can be caused in a human being or an animal when these kind of GMO molecules are consumed. One more guidelines or advancements or regulatory requirements is this WADA, that is World Anti-Doping Agency. It was initiated in Canada during the time of International Olympics, and these guidelines give us more requirements or the requirements for the drugs which are used in sports and are having prohibited drugs in them. This NOPS or Naturally Occurring Prohibited Substances Act also give us the same guidelines which are mentioned by these WADA guidelines, and here they have clearly given the contaminants or cross contaminants or the substances which cause this this sedation or this increased adrenergic activities and all. So they have clearly given substances also which can be naturally occurring or it can be contaminated with the synthetic molecules also. So they have given guidelines on these substances like caffeine, theophylline, theobromine, scopolamine, atropine, codeine, morphine, bufopentanine, and hordenine like this. There are a lot of other listed substances which are given by NOPs for avoiding or controlling of the presence of these materials in the herbal materials. One more is this melanine and related compounds, and there was a big issue also related to this one. I will put it in the next slide. Like this is the analyte specificity, why we have to use more sensitive instruments or more advanced techniques to identify or to exactly know what we are analyzing. Because this melanine, there was an incident in 2008 when people were using this Cheldahl technique for estimation of protein and just converting into the energy values or calorie values. And we know proteins are made up of this nitrogen material, nitrogen compounds, and Cheldahl will easily estimate the organic nitrogen which is present in the protein. But people, for their beneficial effects or for increasing their profitability, they used to contaminate or they used to spoof these food materials with this melamine and related compounds like cyanuric acid and melamine, amylin and amylite, which is having plenty of nitrogen atoms in that one, and it gives the 10 times value of the protein which is there in the food. And because of the consumption of these melamine related compounds, there was a lot of carcinogenic effect which was there in the during the food crisis in that period. And when we are using these many-fold food testing methods, they lack specificity, and often we like these surrogates when we are analyzing proteins with respect to these melamines. It is very much difficult for us to analyze. So we have to be analyte specific, or this advanced technology must be used for this separation and identification of these contaminants. So when we are talking about these challenges of the instruments and their sensitivity, the basic technique or basic requirement for any molecule or any challenge or the major challenge what we face is this harmonization or improving the concentration of the lower level analyte. And in this scenario, there are a lot of extraction techniques or concentration techniques or sampling techniques which are coming to the picture now. And the simple best method which is available is this Squeezers technique. This Squeezers is nothing but a merger of these letters called as quick, easy, cheap, effective, rugged, and safe method. And this technique is now catching much more popularity because of its versatility in application for a large number of compounds, and the combination of salts what we use for cleaning and processing are wide range is there. So we can apply it for major amount of samples for concentrating the samples or analytes. Simple procedure here I have explained. We have to homogenize the sample initially. And by extracting the suitable solvent, we have to, by adding the suitable solvent for extraction, we have to extract the analyte of interest. And by adding the Quecher salt into this one, we can just separate the required analyte from the other interferences, like it may be in color, it may be an organic compound, or it may be in some oils or fats, or it may be in sugars or other related molecules. So our main analyte of interest is separated, and we can get a clear liquid for injection. This can be considered for many other applications like GC-MSMS, LC-MSMS, ICP-MS, or IC analysis. One more example I have given here by using the Quecher's technique. Here we can see spinach, which always poses a threat of this tint formation of green color. We can never achieve a clear sample and sample liquid by using his normal techniques. But when we use Quecher's technique, here we can see a clear liquid can be separated from the mixture of that tint material or any other contaminants in that one, and a noiseless baseline can be obtained when we are using it in more sensitive instruments like LCMS or GCMS. One more sampling technique or sample preparation technique is this solid phase extraction, where by using this is one more, this is like extrapolation of the column chromatography, where we are collecting a fraction of where we are collecting a concentrated or required fraction and once again separating that one. Here we are using the same technique in a much smaller way and to get a better sensitized or better concentrated samples here. Simple experiment I have given here, like when we are using these samples, there are two different molecules like these squares and spheres, triangles and spheres are there. Which is not required is triangle. So when we use this adsorbent material, which is similar in nature or chemistry with this circular material, the circular material is retained and the triangular materials are just eluted out. So once when we pass or concentrate this sample continuously, the adsorbent will be completely saturated with the required analyte. And after giving a suitable wash for removing the interferences and all, this material, whatever is adsorbed onto the adsorbent, is collected into a different solvent, and the complete concentrated analyte is injected for the better accuracy and reproducibility of the analyte results. One more experiment or one more major detector what we are using here is mass spectrometry detector. As I told you, this can be coupled or hyphenated with many number of earlier instrumentation, separation and identification instruments like GC, HPLC, TLC, everything it can be coupled to, NMR also, IR also. And this is a very much popular instrument to get all kind of profiling of the systems, including starting from separation, identification, and characterization data also. Lot of techniques are now available, and latest one which is getting improvement is this multi-TOF. TOF analyzer is coming into major picture now. And as I told you, this mass analyzer, there are a lot of applications, and one more important application of this mass detector is this ICP-MS, which we call as inductively coupled plasma mass spectrometer. And there are different versions of this mass spectrometer, like atomic absorption mass spectrometer, ICP-OES, that is optical emission mass spectrometer, and that is ICP-MS, that is inductively coupled mass spectrometer. These three versions are available. These three versions of this mass spectrometer are available based on the requirement and the data what we are supposed to collect it. And these instruments are all related, except in their sensitivity wise, where we can use ICP-OES or AAS for large amount of samples to be injected, and where we can use ICP-MS for more amount of elemental data to be collected in the herbal material. These heavy metals are always considered as impurities when they are present in a higher level, and the major impurities are like which are considered as big four, or which is given as big four in the United States pharmacopoeia, are this arsenic, lead, cadmium, and mercury. And this is one more sampling technique which we are talking about is microwave technology. And as we know, these normal samples we cannot just directly solubilize and inject it, because these molecules are not very much freely available as such. So we have to break the entire matrix of that material, which is not possible in the simplest way. So in an advanced way, in a simple way, we can use just an normal hot plate to acid digest the sample and use the ash which is obtained, which is having these all concentrated minerals in that one for analysis. But that always gives the repeatability problem, and other contaminations is always possible. So much better advanced technique what we are using here is this microwave digestion technique, where we use limited amount of sample, limited amount of solvent which is added to that one, and which is heated at a very high temperature and under high pressure conditions, so that the entire matrix is broken down and the metals which are present are converted into their nitrate forms, for the better absorption and identification of the metals with easy quantification also. These heavy metals are relatively dense materials noted for its potential toxicity. So the removing or reducing heavy metal content in the herbal extract is a very big challenge. Moreover, at the production level, if we control also, the heavy metals can be entered into the material during the handling procedures also. So we have to put an high alert on this heavy metal contamination from the agricultural field till the consumption. So here I have given few guidelines about the heavy metal requirements. As per the AICTE and LATAM requirement, lead is 10 ppm, cadmium is 0.3, arsenic is 3 ppm, and mercury is 1 ppm. So here we can see this cadmium, arsenic, and mercury will be almost like same for almost all the countries, but it only changes with respect to lead and mercury for few of the countries over here, where this American guidelines is having 5 ppm of lead as a limit, and the European guidelines based on the category of the medication what we use, like if it is used as a medication or as a food supplement or dietary supplement, it depends on the different levels of the arsenic to be present in there. And coming to much more advancements of this heavy metal analysis using ICP-MS technique or hyphenated technique, this speciation is one of the important criteria now which is picking up. As I can give you one example of this monomethyl mercury, which is present, because mercury we are analyzing it in a single form, but we don't know how many different organic forms or inorganic forms mercury might be present in the sample. So when we come with respect to monomethyl mercury or methyl mercury is the one which is more predominating than dimethyl mercury or monomethyl mercury. And this presence of or conversion or availability of this monomethyl mercury depends on the acidic or neutral conditions or aerobic or anaerobic conditions. Recently, we had a crisis in Kerala, I think it is in 2016-17, where people were identified with lead poisoning. And when it was researched or when data was collected, they identified that people who are living nearby the sea coast or people who are consuming more amount of fish are having this problem, because in anaerobic conditions the presence of this methyl mercury and all is more, and in acidic and neutral conditions it is very much different. One more problem is the presence of this chromium in a different oxidation states like chromium 6 and chromium 3. In aerobic conditions, chromium is present as +6, and in anaerobic conditions, it is present as +3. +6 chromium state is always considered as an toxic material or a carcinogenic material, whereas +3 condition is considered as a trace nutrient. So we have to able to identify whether chromium is present in +6 state or in +3 state when we are putting it as on a label or under our or we have to control these kind of conditions in our finished products also. So to establish these kind of speciation where we can separate +3 from +6 of chromium or monomethyl mercury from dimethyl mercury, we have to have a highly sensitized instrumentation wherein we are using the hyphenation of ion chromatography with ICP-MS or liquid or HPLC with ICP-MS. So these kind of hyphenation is always, or advancement is always required for the registration requirements or regulatory requirements. One more use of this hyphenated technology for speciation analysis is like separation, excitation, and detection. So we can use HPLC coupled with ICP-MS, GC for ICP-MS, or ion chromatography with ICP-MS, wherein we can achieve this separation, excitation, and detection. One more example I would like to give here is when we simply inject arsenic or the normal form of arsenic, which is digested in microwave digester using nitric acid as a solvent, we can have different forms of this arsenic which can be present here. That is in the form of cacodylic acid, or in the form of arsenious acid, which is As+3, or which is arsenic acid, which is As+5, or which is MMA, that is monomethyl arsenic acid. So here when we recall with respect to limit tests of arsenic, we can just we know the principle that it is just converted from As+3 to As+5, from arsenious acid to arsenic acid, and it reacts with that material to give an yellow color, and we are just comparing the intensity of that yellow color which is produced. But here when we see we are using an separate ion chromatography coupled with ICP-MS, we are seeing 4 different forms of arsenic which is possible. Here we can see everything is almost like resembling only, except for this CH3 or CH3 or double bond O or OH only. So here the molecular weight of the arsenic acid, the different species of arsenic is different, but when we see it as a whole, it is resembling only arsenic. But individual forms are having individual toxicity levels, which has to be controlled. So this kind of speciation has to be determined when we are going for heavy metal analysis or going for establishing the toxicological data for our new molecules. So these kind of advancements and hyphenation will always help us in achieving the regulatory requirements. One more thing in this markers analysis for food, I would like to add some points. Still we are working on this now. So presently I have small information on this one, where we are using these isotope ratios. This technology is called as IRMS, or isotope ratio mass spectrometer, and this is the most powerful markers for food classification, and a fact for certified by several hundreds of scientific publications are there with respect to these isotope ratios, where we can, this is nothing but an advancement of mass spectrometer, where we are having two molecules, where one molecule is targeted and analyzed. But what about the other molecule which is present, or the small isotope which is present for that major targeted molecule? So these kind of markers are also targeted in this IRMS, and much more data or availability is possible by using this isotope ratio, these things. Present applicability of this material, here this video will be going on during my talk also, so you can just have a look at that video. The basic application of this IRMS is, in one major thing is this differentiation between these natural and synthetic aromatic substances, where this benzaldehyde from natural material or from synthetic material or synthetic origin can be easily identified and analyzed by this IRMS technology. One more application is the analysis of this natural and synthetic caffeine, where we can differentiate whether it is from the plant source or chemical source by marking the isotopes of carbon and oxygen. One more important applications and presently picking up applications is distinguishing the agricultural and synthetic practices. As I told you, a lot of these NOPs, NOPS, WADA and all are talking about the contaminants or prohibited substances in our molecules. So we don't know whether it has been contaminated from the synthetic origin or from the agricultural origin. So it can be, this IRMS technology is highly useful in differentiating those kind of molecules also. And one more important application is this fraudulent honey which is used. So we can know whether the sugar is from natural origin or from synthetic origin. So these are some of the few applications here for this IRMS. And I have a much more better video for understanding of this IRMS technology. I will show it in the last. So in this slide, I have given some of the limits which are the requirement for the major regulatory guidelines as per and GMP practices also. And in my earlier slides, I have already given these details, so I will just skip these things. And here I have given the example of the Maggi, where we all know that it was a big issue in the food industry because of the presence of this Ajinomoto, or what we call it as MSG, that is monosodium glutamate. So when Maggi was analyzed with a normal instrumentation and with our normal FSSAI technology, means like limit test kind of things, it was passing. But when the same was injected using ICP-MS, there was a big issue that it is crossing the toxic limits. So we are all aware of that Maggi issue. And one more thing is this Haldiram's issue, and that person also was jailed for some time because of this issue, because they are using nitrogen for packing these snacks and snack materials, which caused this salmonella bacteria or salmonella species bacteria to develop inside the snacks. And because of the consumption of this one, a lot of people got their health issues, and now it is banned. In US it is banned. Haldiram's is not available in US because of this nitrogen packing issue and salmonella typhi issue. And this whatever I have given here is that wheat gluten. So I think we can recall the issue of ITC, Ashirvad Atta, where there was a big issue, and in WhatsApp and Facebook and all, everywhere we have seen videos about this gluten content, and a lot of people have shown that they are adding plastic materials into the Ashirvad Atta, but that is nothing but a gluten content which gives the elasticity to the Ashirvad, the wheat flour, but the amount which has to be present inside that material has certain limitations. So there is a lot of work going on in this aspect also. And now Ashirvad has come up with gluten-free atta itself. So no issue of plastic over there. And one more thing, one more example of this analysis is this: when we are skipping out some sensitive information, the chocolate, what I have shown here is Cadbury's Dairy Milk chocolate, which is having that nuggets or worms which are coming out from the chocolate after some time, because during the processing of this chocolate, it was not taken care, or it was not sanitized procedure was used. So when we are using well established procedures or sensitive procedures or advanced techniques, these kind of process errors or presentation errors can be avoided. So these are some of the issues which have already been addressed with the advancements which are present, and all these advancements are applicable for our herbal materials. So here I have presented these few videos. Maybe it will be helpful for you, and these are all available in YouTube also. So for further requirements or any further clarifications, you can just visit that website also. So this video is regarding the Anton-Par multi-wave GO system, which is used for microwave digestion of the herbal materials. And this is, I would like to say, this is a very much compact model presently used for high throughput sampling, where we use minimum amount of sample of 100 mg, and the sample quantity can be as low as 1 ml. So at a time we can process around 32 samples in this one within within like 15 to 20 minutes time, and to the complete dissolved state. This easy handling of the Anton-Par tubes, the multi-wave GO tubes are very much beneficial, because they avoid minimum damage to the parts what they are used, and like replacement costs will be very much minimal. The samples are placed, added nitric acid in that one, close the lid, and we are closing the microwave digester system, so which gives us complete safety from the high pressure and high temperature which is built up into the microwave digestion. So we have the pre-programmed methods available, or based on our requirements, we can program the conditions, or means like we can increase the temperature, or we can increase the pressure, or we can increase the amount of time exposed for better clarity of the solution, or for better digestion of the elements which are present in the molecule. So within 18 minutes, 12 samples are digested using this microwave digestion system. So that's about the micro digestion system. And one more presentation is there, one more video is there with regards to the Quecher sampling technique. So this video is self-explanatory.
Traditional methods can be costly, multifaceted, and time-consuming. Plus, different sample types require different methods, which can be complicated, laborious, and add to the cost. Catchers is a simpler technique. It's simple to implement and cost-effective and provides high-quality analytical results. Catchers stands for quick, easy, cheap, effective, rugged, and safe. The Catchers technique allows you to prepare a large number of samples in a short period of time due to the simple process employed. This, in combination with the low volume of solvent used and lack of chlorinated solvents, helps provide an inexpensive method. The technique allows the targeting of a wide range of compounds from various food samples and provides recovery and reproducibility levels in line with industry requirements. Typically, endpoint analysis is performed via GC or LCMS. Here's how Catchers works. First, homogenize the sample by grinding it as much as possible to increase surface area. Break down cell structure and ensure the analytical test portion is representative of the whole sample. It's often important to keep the sample cold to prevent analyte breakdown. Cryogenic milling is recommended. Next, add the internal standard and the extraction solvent, typically acetonitrile, into the centrifuge tube with your sample. Shake vigorously, then add the extraction salts and buffers and shake again. For this step, you can use thermoscientific prepackaged salts and centrifuge tubes. Then, centrifuge the tube to separate the organic layer and transfer the organic layer to another centrifuge tube for the next step, cleanup. In this final step of the Catcher's workflow, add DSPE or dispersed SPE to the tube containing the organic extract. The DSPE step reduces the concentration of fat, proteins, chlorophyll, and other undesired matrix components. You can use thermoscientific dispersed SPE for this step. Shake again, then centrifuge again. Then transfer the final centrifuged clean extract from the tube to a sample vial. Your sample is now a fully clean, particle-free liquid, ideal for analysis. Some important tips and recommendations. Number 1, if you must homogenize at ambient temperatures, work as quickly as possible and mix continuously when withdrawing your test portions. Number 2, for dry samples like cereal, tea, and spices with water content less than 25%, you may need to hydrate by reducing sample size and adding water prior to adding solvent. Number 3, during solvent addition, an internal standard solution can be added to monitor recovery and aid quantitation of target analytes. Number 4, different versions of Catchers use slightly different amounts of extraction salts and buffering reagents. Generally, salts are present to aid particulates from the aqueous to the organic layers, and buffers help to control the pH, protecting sensitive analytes. Number 5, different versions of Catchers use different dispersed SPE sorbents also. Typically, PSA is mixed with C18 for fatty samples, and PSA is mixed with gravitized carbon black, GCB, for foods with high levels of chlorophyll or carotenoids. Other combinations of cleanup products are available for a wide variety of applications. Number 6, some pesticides, like those strongly protonated at low pH, require pH control and need to be buffered to pH 2 through 7 prior to extraction. Number 7, after sample preparation, acetic or formic acid can be added at low concentrations to ensure stability of base-sensitive pesticides. Analyte protectants like sorbitol, glutatolactone, or epiglycerol are sometimes added prior to analysis by gas chromatographic techniques. In summary, the Catchers method has revolutionized sample prep and cleanup and has become the industry gold standard. Visit www.thermoscientific.com/pesticides for more on Catchers, including method details and downloadable webinars and application notes.
So one more small presentation about this IRMS, that is isotope ratio
Now
MS.
suppose we throw a lighter and heavier ball in a linear direction, then in normal condition, those balls will go in a linear pattern. But if we will apply side force, then this ball will deflect. and that deflection will depend on the masses of the ball. Like lighter ball will deflect more than the heavier ball. So IRMS also works with the same principle. Like here I am showing you 3 parts of the
So here I have just given the full details, means like there is a video available on what is the application and how the exact instrument looks for IRMS.
Now here I would like to show you the main instrument, and I will provide information regarding the fundamentals of isotope ratio mass spectrometer. So now this machine is the isotope ratio mass spectrometer, and this is a model MET 253. Now this model has a 3 parts. One is source, second one is magnetic field, and third one is collector. Okay, so this mass spectrometer is a gas source mass spectrometer. But from field, we collect solid rock samples or sediment samples or water samples. So those samples we cannot put directly into this instrument. So for this purpose, basically we have a 3 peripheral parts. One is gas gauge, second one is GC isolate, and third one is elemental analyzer. So now with the help of these 3 periphery, we produce the gases, and then those gases we send from that part to IRMS. And this IRMS measures the isotopic ratios of desirable gases. Now basically the sample.
Now in this video, next video, we can see how the sample is getting prepared for the IRMS analysis.
So first we dry all those samples and we take homogenized 1 or 2 milligram, 1 or 2 gram samples, representative samples. Now for organic carbon isotopic analysis or hydrogen isotopic analysis, we crush the sample and we make a fine powder. And for carbon isotopic analysis, we treat those samples with 5% NCM. And then after 3-4 times acid treatment, we again wash those samples with the help of milky water. And after that, we dry those samples and then again we crush those samples to make a fine powder. After making a powder, we first of all we pack those samples into the tin capsules. Why we are? Because along with carbon isotopic values, we want to measure total organic carbon content. For this purpose, we have to weigh our samples. So now these samples we have packed in tin capsules, and then these tin capsules are introduced in the elemental analyzer. Through the flux combustion, all organic content converted into CO2 gas. Those CO2 gas passes through the reactor, water removal trial, and column, and finally that gas reaches to the complophore. And from complophore, that gas enters into the mass spectrometer, where this gas ionized, entered into the magnetic field, and then ion particles are collected in different cups.
So this concludes my presentation, sir. So if you are having any doubts or any clarifications from my side, I can take the questions now. So I will be able to, if I am able to answer now, I will answer. Otherwise, I can always come back to you. So I'm open for the questions or clarifications. Thank you. Thank you very much for all those patients listening.
Session is open for the question and answers. If you have any queries, please do feel free. As sir has already agreed, it would be nice if you can ask questions. That will be fine. Or else even you can get the clarifications so that we can pass on your questions.
Good afternoon. This is Dr. Sholak from KLE College of Pharmacy, Hubli. Thanks for enlightening us with the latest analytical techniques. My question is on TLC-MS method. If the compound is non-EUV active, then we go for derivatization and RF value calculation. Can those compounds be subjected to MS analysis?
Definitely, sir, because we have coupled this TLC analyzer with both the densitometer as well as with mass detector here. In my example, in my slide also I have shown the same thing. So when we are using this densitometer, we can directly scan the TLC plate in both the UV and visible ranges both. So you can derivatize, or without derivatization also we can directly scan the molecules. So always whatever the spots of interest are there, we can subject it to MS analysis.
Okay. If the compound is derivatized, I think mass of the compound also changes. And what will be its effect in our
That's what, sir. After derivatization, now we are aware that molecule where it is exactly there, right? We will be identifying the RF values of that molecule of the spot. So before derivatization itself, we can just check for that RF value in the densitometer, and we can subject the same spot to the MS analyzer.
Okay. Thank you, sir.
Any further questions? I think all the participants and even organizing committee people have explored the power of listening, and I'm sure that you also might have explored the power of sharing the information. I have a couple of thoughts that gold has got its own value, and similarly stones and gems have got their own value. But when you join together, it becomes a very good ornament. So we were lucky enough to have both of you from Himalaya for this particular morning session. And on behalf of the KLE College of Pharmacy, on behalf of the organizing committee and AICTE QIP program, we extend thanks to Dr. Rajendra B and Dr. NS Prakash sir. And we hope that we will have the continued support in future also. And we'll have an opportunity to listen such lucid lectures in future also.
Thank you. Thanks for giving us that opportunity.
Thank you, sir. Thank you. Thank you, sir.
Thank you. Thank you,
Rajendra sir. Rajendra sir, from your presentation, we come to know that the academics are nearly 1 decade behind that what we have in our labs and what is there in the industry. So I request please have industrial institution collaboration so that our teachers and our students will be trained for this new instrument, sir. We are looking forward
sure, definitely, sir. Already we have. Yeah, in that work already we are working, sir. Himalaya is collaborating with a lot of institutes and organizations, academic organizations for these kind of activities.
Okay, thank you, sir.
Okay.
Thank you.
Thanks for all the participants. Okay, for patient listening.